Great. Thank you, Swati. And it's really a pleasure to be here. As I said, my name is Sanjiv Shah. I am at Northwestern University and the principal investigator of the Data Translation Center for HeartShare. So I was asked to give our vision for AMP heart failure. And really, I'm going to give you the vision as the PI of the Data Translation Center, as a clinician and a researcher who works in this space and takes care of these patients day in and day out, and having really collaborated with all of you. So what I thought I would do is talk about what it would look like if we're successful, and hopefully we are with all of your help and others, how we would manage HFpEF in 2030. So that's 8 short years from now. So, not too far away, but I think by accomplishing this project successfully, we can get there. So we have a breathless patient. Now, this patient may come into our office as an outpatient, just very breathless, and they exercise intolerant. They've never been hospitalized for heart failure. They don't have any fluid on their exam. Or it could be someone with a prior diagnosis of heart failure, a prior hospitalization, but there's some questions about the phenotype. Regardless of the type of patient, they're breathless, they come in. And we have 2 levels of data assessment. So first of all, we get an EHR data download in a very structured format that facilitates our review of that patient. We get an ECG and an echocardiogram, and we get blood and urine samples. Now, sometimes that alone, we can make the diagnosis. Say they've been hospitalized, they have an elevated BNP, and their echo is suggestive. But many times we're going to level 2. And here, maybe we're doing things differently. We're having them wear a monitor that gives us a ton of data about what's going on in their actual lives. We have exercise phenotyping, some that we, of course, in the HFpEF field are really trying to advocate for, more exercise phenotyping. I see Greg Lewis and Barry Borlaug and others who do this a lot. And then stool and saliva samples, which we are not getting right now, but which could give us great information. And then we take these and, can I have control again? It was working beautifully. Okay, let's see. Yes. Okay, so very important. There's still going to be a clinician review. We are not going to be outpowered by AI in the next 8 years, hopefully. But that clinician review is really important. And let me give you an anecdote. What I've been advocating for years for in trials, HFpEF trials, is that a lot of patients have heart failure and they have a preserved ejection fraction, but they don't have HFpEF. They have constriction, they have amyloid, they have other things going on that are not really captured by clinical trial inclusion-exclusion criteria. Even though we put them in there as words, the clinician has to do it. And oftentimes we're blindly, I think, and I've suffered from this, we're just sort of like, I need to get patients in the trial, We overlook things. And so I've been advocating for a screening committee, a clinician review, an expert clinician review. And we finally are doing that with some device trials, and it's fascinating. We have expert clinicians. Even at my own site, I basically, gave myself a red flag for trying to get someone in the trial. One of my patients, and when he got in, it went to the screening committee, and I myself realized that this guy had a Brady arrhythmia. He has HFpEF, but he has a Brady arrhythmia. And I treated him with a pacemaker, and he got a lot better. And I was trying to get him into a HFpEF trial. So there's things in the screening committee, and we have a well-oiled process where they get an echo, they get the baseline data, it goes up into the cloud. We have 4 reviewers that are experts in HFpEF. We review it, and we actually figure out what they have. We say, are they eligible or not? We found amyloid, we found constriction, this patient of mine that needed a pacemaker, severe aortic stenosis. So sometimes having that external clinician review, that expert review, is really important. I think it'd still be important in 2030. We're going to have additional testing. Maybe we need to get a cardiac MRI. Maybe we need a PYP scan. We're going to have an omics analysis, automated image analysis. Ultraomics will be doing that for us, and it'll be part of the process that we all use. And then there'll be ML-based classification. We, in the heart failure AMP program project, we will have automated algorithms to classify patients. And then what we'll do is we will have a phenotype library. So we have these discernible phenotypes. They may be based on omics, they may be based on images, or a collection of these things, but they're prototypical types of HFpEF. And maybe this particular patient doesn't fit that bin and it's a new type, but we have this phenotype library that we can assess. And we use that to target patients. Now we know in 2022 that the first line of therapy for the vast majority of these patients, the proven therapy is SGLT2 inhibitors. But then this is where we're going to take targeted therapeutics, and maybe we'll be there, maybe not for all of these, but there may be a non-steroidal MRA, and we say, okay, this phenotype really benefits from this. Maybe it's an ARNI, maybe it's a myosin modulator, maybe it's an antisense oligonucleotide, and many more. We're just talking about the companies represented here, but there's so many therapies that are potentially available. And this is the vision for 2030, but it doesn't stop here. We're doing that multi-level testing. We've got all the clinician review, additional tests, omics, ML, and we get this phenotype library. We do the targeted therapeutics. Are we done there? No, we're not, because the whole goal here is to continuously learn. We're going to have outcomes assessments that are standardized. We have the Eureka app here that we're going to hear about later today. And the Eureka app is really amazing. It really helps us with the patient. Everyone has a smartphone, or most people do. And they have the smartphone, they get, and they're geolocated near a hospital or ER. And if they're there for 3 hours or 4 hours or more, they'll get a ping. Are you in the hospital? And so we're really trying to engage the participants through their smartphones. So ongoing EHR laboratory outcome data collection. Now we can refine the machine learning model. Now we can evaluate for bias assessment. We heard a little bit about this before, but this is a huge issue, bias in machine learning models. And then this then adds to the HFpEF phenotype library. So it's this continuous learning, continuous refining, pattern and trend recognition. One of the really interesting things is that in the 1970s and early 1980s, where HFpEF was first recognized, it was really a syndrome of hypertension, of LVH. And as we learn more about it, as the decades went on, we now think of HFpEF as obesity metabolic syndrome. We kind of downplay LVH and hypertension, even though that still exists. And so we have to ask ourselves, is this because we just were not too smart in the 70s and 80s, or have things actually changed? And I think it's really the latter, that things actually change over time, that syndromes like HFpEF evolve over time. And it's just like the sort of evolution epidemiologically of the risk factors. We see different types of HFpEF over time. So that last, the trend recognition, is going to be something that's also important as we think about HFpEF and the vision for 2030. So how are we going to get there? How are we possibly in the next 5 years going to propel ourselves towards that? And besides all of the industry partners, the NHLBI, the FDA, the associations, the heart of the AMP program that we're talking about here is HeartShare. In the afternoon, we're going to be going in detail, but that's where the Data Translation Center at Northwestern and our sites here that have been really actively engaged in this program, and others. We're bringing many other people into the fold with ancillary studies that are currently being evaluated now. But these are the participating institutions. And we saw a little bit about this, but just so we're all on the same page, the study design overview. Remember, there's that retrospective, the extant data sets. We're going to hear about that. I'll talk about that later today. This is really the key resource for our industry partners in 2023. So this is the thing that we're going to work really hard on building next year, so that you have access, that we all have access to this data, omics, and images, the raw images. That then feeds into how we do the prospective part of the study. Now, obviously, we're planning it now, and I'll talk about where we are with that, but we are going to be enrolling our first patients in a few months. So, this is something that is going to be continual learning from this retrospective part to modify, add on to, and help drive this prospective portion of the study. And then we talked about the master protocol that'll get us to 2030, hopefully. Now, this is the scientific approach. And, it's really about harmonizing all the data from these prior studies, getting them into this Biodata Catalyst portal, and then two types of approaches. And I think we've talked about this before, but I think it's really important for companies to understand, and our industry partners, associations, nonprofits, that there is still going to be a hypothesis-driven approach. We know many discrete HFpEF phenotypes exist. We're already studying them. But now we can leverage machine learning and omics to do all kinds of things with it. We can identify what are the molecular drivers. We can use machine learning to auto-identify some of these phenotypes, and I'll give you an example. And then, of course, the unbiased approach, which we say, what do we know? Let the machine figure it out with unsupervised learning. We can also think about a molecular-driven approach. If one of our industry partners or one of our academics has a certain pathway that they think is important, we can now have this resource where you can interrogate that pathway and see, okay, which patients have that pathway upregulated? Which patients, what is their phenotype? Can we auto-identify it and learn more about it? And so everything here gets into Biodata Catalyst, and that's where we access the data. Okay, so also to drive home the point that we have 3 levels of data collection at our 6 clinical centers. So it's this inverted pyramid. And so I really want everyone to be on the same page about this. The data portal committee is also going to talk about this. Our Eureka, we're going to talk about this. But we are getting EHR data and hopefully images from every single heart failure patient that has touched any of the health systems involved. And these are very large health systems. We're going to see some numbers later this afternoon from 2016 onwards. And we have some very specific criteria that we think increases specificity. And why do we want all of heart failure? If this is a HFpEF focused AMP, is because HFpEF is what we're trying to solve, but there's all other problems in heart failure. And we want this captive group that can feed studies going forward. And so we were hoping for over 100,000 heart failure patients. You're going to see it's way more than that we're going to have access to their data. So that's level one. Level two is a subset. Every one of these patients is going to be asked to participate in what we call a low touch heart share registry, which is totally remote. It's this through Eureka, this smartphone and web-based app. We can also have coordinators call patients and get them in so that it's not all people that have the means to access the web and have smartphones, but they're going to integrate with this Eureka app, and we're hoping to get 10,000 people in there. And they are going to be, they're going to be active participants in the app, and we're trying to figure out the right balance so they're not annoyed by the smartphone pinging them every 5 seconds, but also engaged. But these engaged participants for our industry partners, they're great because we can just send a blast, say, hey, we have a study. If you're interested, click this button. All of a sudden, within a week, we have 100 participants. This is a great proof of concept. And then finally, we have the deep phenotyping protocol that we're going to hear more about with Barry's presentation this afternoon. And that's really the deep phenotyping prospective study that we're talking about at the 6 clinical centers. Right now, it's 750, HFpEF 250 comparators, but we're hoping to increase that to up to 2,000. Okay, so let me give you an example of how we can use this resource for diagnostics and drug discovery. So, we really want to use unbiased discovery to augment what we are doing with hypothesis-driven discovery. And this is just one example that we've been trying to use. So we have echocardiograms, we have the raw DICOM data, and we can use an autoencoder to basically use deep learning to take this raw image, which are a bunch of pixels, that's a video, and basically deconstruct it into data. So basically have the computer grab data from this, try to distill this complex image into a discrete set of variables and reconstruct this image, and then compare, does this image look like the first image? And keep doing that until it feels confident that it's got the right set of data that is explaining what this image means. We can then take that data and combine it with the omics data. We reduce that data in a number of ways. One is simple, one is principal components analysis. Now we have a usable amount of data that we can cluster together to find phenotypes. And like was presented in the Ultromics talk, we have saliency maps that can tell us where the deep learning model is potentially focusing when we find these different categories, when we try to identify them. What is it focusing on? So this is an example from David Oyang's lab where they just try to figure out the age of the participant. And the computer knows nothing else. It just knows the training data set has age and the echocardiogram. And where does it focus? Well, it focuses on the mitral annulus, which makes sense because the mitral annulus classifies with aging. And so something similar to that, and what we saw in the Ultromics presentation, where is the computer focusing on? That can give us clinicians and researchers additional insight. So it's this type of machine learning with multi-omics that we hope to incorporate. And this is just one example. But then there's also phenotype specific discovery. And this is a prototypical example. I would say this in 2022, an easy example. You look at these two echocardiograms, garden variety HFpEF and cardiac amyloid, and you don't have to be an echocardiographer to say, these are very different. And yet it gets missed all the time. And yet computers for sure can spot the one on the right. And so can we train a computer to identify other types of HFpEF, other phenotypes that we discover, either because we already have identified them, like obesity, low BNP HFpEF phenotype, or something that is tied to a molecular pathway that we haven't discovered yet. And we've done this. This is out of Rahul Deo's group at Brigham and Women's, where we've taken deep learning to identify cardiac amyloid, and you can see the AUCs are getting up to greater than 0.9. So really good, a computer is getting really good at figuring out what cardiac amyloid is. And I know many of the industry partners that do this work, like Ultromics, are working on things like this. This is helping auto-diagnose these diseases like cardiac amyloid. Why couldn't we do that with other forms of HFpEF? And we did this study in collaboration with Pfizer, where we said, a lot of times patients at risk may not have the echocardiogram, or that's not available, but EHR data is, medical claims data, ICD codes. So we used huge data sets like IQVIA and Optum to try to figure out what diagnostic codes are associated with amyloid. We can do the same thing with HFpEF subtypes. And so that gives us a predictive algorithm. We can prospectively validate it, which is what we're doing right now with this amyloid algorithm. But it gives us something to say, okay, your probability of having a specific type of HFpEF is higher in this individual patient, which then triggers the clinician to go about trying to figure that out. And the other thing in light green in the middle is that you can look backwards in the EHR and try to find out what antecedent risk factors are there. And we did this, I don't know how well you can see this, but this was really a proof of concept. So the first thing on the left is carpal tunnel, and it goes back 10 years. So 10 years prior to the diagnosis of cardiac amyloid, patients are having carpal tunnel. And we know that, clinicians, we figured that out. But there's enthesopathy, synovitis and tenosynovitis, sensory disturbance, spinal stenosis. The cardiac stuff on the bottom happens late. So now we can identify clues. These might be things that are related to the pathogenesis of disease that we identify. Okay, so let me drive this home. Last few slides between me and lunch, or you and lunch, all of us in lunch, is phenotype specific discovery. Okay, so it looks like this is not playing, but we have deep learning for HFpEF subtype identification. So this is a 74-year-old woman with HFpEF, BMI of 46, BNP of 28. So obese, BNP deficient subtype. And you can see severe pericardial and epicardial fat over the right ventricle. And if this played, you would see that the septum was bouncing, and Barry has described this, and we see it clinically, that this is the prototypical obese phenotype. They've got this, basically they've got a pericardial constraint, their volume expanded because they're obese, and that blood can't get into the heart because they have all this fat encasing the heart. And so identification is really important, but I have several obese patients that don't have this phenotype. They have something totally different. And I have patients with a BMI of 30 that have just intense visceral adiposity. We see that a lot in South Asians. And so, how do we treat these patients? So this on the bottom right is a patient of mine with this type of phenotype. And in 2017, June, I started a GLP-1 receptor agonist. BMI was 39. She was class 3 on a ton of medications. And by December of 2018, she was class 1 off of all antihypertensives and all diuretics. Now, we may not see that kind of cure, but you can see how there is a therapy potentially that's being tested right now that could have a major impact on this type of patient. So what can we learn about the AMP-HF, about specific phenotypes, taking to mind this obese low BNP phenotype? So let's say we have this resource that we're trying to build, and we're looking at this specific phenotype. Multi-omics analysis of the blood and adipose tissue. So we can now discover underlying molecular pathways that could be targeted pharmacologically. Maybe there's something on top of weight loss that we can specifically target that melts away pericardial and epicardial fat. There's advanced image analysis, MRI tissue characterization. I don't know about you, but I have patients who have a lot of epicardial and pericardial fat, but it glides and slides against the liver. It looks beautiful. They don't have this ventricular interdependence. They do seem to do just fine. And then I have others where it's very fibrotic fat, and it really acts like a constraint on the pericardium and on the heart. So fibrotic and stiff fat from malleable spongy fat. That's advanced image analysis. EHR data mining, kind of like we did with amyloid. Let's look back 10 years. Is it just obesity, or are there other things going on? Do they all have rheumatoid arthritis? Is there some inflammation going on? Is there something that makes a patient susceptible to this? Automate the diagnosis. So deep learning of ECGs and echocardiograms to identify the diastolic septal bounce, epicardial, pericardial fat. One of our HeartShare research fellows is working on something like this. And we can apply EHR tools and deep learning algorithms on the images in the health system to find these patients automatically and get them into trials. So this is really what we're trying to do. So I'll end with the key deliverables. And I've showed you many of you these slides before, but I think it's worth revisiting. Our goals with AMP-HF and the HeartShare program are to create a rich repository of data, omics, and images from multiple large-scale epi studies, multi-center heart failure clinical trials, other registries, And really, we want to facilitate open data science for the investigation of HFpEF. We're building this. It's going to be exclusive to us for the time we're here, but ultimately, we've got to open it up to the public to be able to leverage all of the superstars, especially our younger colleagues who are really doing important work. We need to define the subtypes of HFpEF and develop algorithms to identify the subtypes, discovery and prioritization of targets and mechanistic pathways. This is how we're going to develop new therapies. We're going to establish a large EHR-based cohort that can be enrolled in registries for future studies. So that's the low-touch registry. And then the deeply phenotyped patients who can support target validation and facilitate proof of concept treatment trials for HFpEF. And finally, as you'll see, we'll get an update from Phil Greenland this afternoon on training the next generation of heart failure data scientists. So we're taking data scientists, teaching them about heart failure. We're teaching heart failure fellows about ML and AI techniques. It's really quite exciting. So I'll end there. Thank you. We're going to have some final words from Tanya and I.